Okay, so chapter 10, muscle. Functions of skeletal muscle. So we're going to use muscle for body movement, so that we can move bones, make facial expressions, speak, breathe, and swallow. Muscle helps us maintain our posture, stabilize our joints, and maintain body position. Muscle also gives us protection and support. It packages internal organs and holds them in place. Regulating the elimination of materials. Circular sphincters are going to be in charge of that. They control passage of material at orifices. And remember that muscle is involved in heat production to help maintain body temperature. When you get too cold, you start to shiver. All right, characteristics of skeletal muscle tissue. I want you to be pretty familiar with these definitions. Excitability, this is going to be the ability to respond to a stimulus by changing electrical membrane potential. Conductivity, muscles are conductive, and conductivity involves sending an electrical change down the length of the cell membrane. They're also contractile. This is exhibited when filaments slide past each other and gives muscle the ability to cause movement. Characteristic of elasticity. This is the ability to return to the original length following a lengthening or shortening. And extensibility, the ability to be stretched, okay? Be very familiar with these terms. One of these terms is for sure going to show up on your test. And so I would like for you to know what each of these mean. All right, so let's talk a little bit about the gross anatomy of skeletal muscle. Skeletal muscle is composed of skeletal muscle fibers, aka muscle cells, okay? In addition to the muscle cells, skeletal muscle is going to contain nerves, blood vessels, and connective tissue. You'll see skeletal muscle cells are organized into groups called fascicles. So here is a fascicle right here. And it's a bunch of individual muscle cells. So this is one muscle cell or muscle fiber. And it's one of many in a fascicle. And a fascicle is a bundle of skeletal muscle cells, as we said. There's going to be some connective tissue components, 3 concentric layers of wrapping, in fact. There's the epimysium, the paramysium, and the endomysium. The epimysium is dense, irregular connective tissue wrapping the whole muscle, okay? The paramysium is going to be dense, irregular connective tissue wrapping the fascicle. And this is where the blood vessels and the nerves are going to be found. And then you have the endomysium, which is going to be areolar connective tissue wrapping individual muscle cells. This is a delicate layer for electrical insulation, capillary support, and binding of neighboring cells. So here we have the muscle itself, this whole organ here. And then surrounding that, we have the epimysium. Then we have fascicles on the inside, arranged like so, with the paramysium between them, okay? And you can see the artery, the vein, and the nerve here in the paramysium. And then you go a step further, you look at an individual fascicle, you see that it's composed of many muscle cells, and in between these muscle cells is the endomysium, okay? And here's one single muscle cell. You can see multiple nuclei. Remember that striated skeletal muscle is going to be multinucleate, okay? You also have a deep fascia and a superficial fascia. The deep fascia is a sheet of dense, irregular connective tissue. It's going to be located external to the epimysium. And this separates different muscles while also binding them together. This is going to contain nerves, blood vessels, and lymph vessels. And then you have the superficial fascia. This is areolar and adipose tissue located superficial to the deep fascia, and this separates the muscles from the skin. So here you can see the epimysium, and then surrounding that is going to be the deep fascia, and this is going to bundle this whole cell, this entire organ, together. The deep fascia covering the muscles, the superficial fascia between the hypodermis and the muscle, okay? Okay, blood vessels and nerves. Skeletal muscle has extensive blood vessels to deliver oxygen and nutrients, as well as remove waste products. And then skeletal muscle is going to be innervated by motor neurons. Motor meaning to move. And that makes sense, right? The neurons that innervate skeletal muscle or innervate movement are called motor neurons. These originate in the brain or the spinal cord and have long axons traveling to skeletal muscles. The axons of the neurons then branch and terminate at neuromuscular junctions, and this allows voluntary control of contraction. And I'll show you what I mean in a second. All right, the microscopic anatomy of skeletal muscle. Well, what are the parts of a muscle cell? You have the sarcoplasm. This is what is the term for cytoplasm in a muscle cell. It's going to have your typical organelles like mitochondria, Golgi, etc., plus contractile proteins and other specializations. Multiple nuclei. Individual cells are multinucleated. The cell is formed in embryo when multiple myoblasts fuse. There's a sarcolemma. This is going to be the plasma membrane of the muscle cell. And it has these things called T-tubules or transverse tubules that extend deep into the muscle cell. And what that does is that allows for conduction of an electrical signal deep into the muscle cell and essentially synchronize contraction of the muscle cell. Okay, so this is showing the outer boundary of a muscle cell. You have the sarcolemma, or the plasma membrane. Several voltage-gated ion channels here, a sodium channel and a potassium channel, help set the resting membrane potential. Recall that the interior of the plasma membrane is going to be more negative than the exterior, as you can see here with these negative charges. But what I really want to show you in this diagram is how the T-tubule is just essentially an invagination of the sarcolemma down into the muscle cell. So this is the interstitium right here. This is the muscle cell. This is the inside of the muscle cell. And this is the muscle cell membrane. And it kind of dives down into the muscle cell itself. Invaginates, and that's a T-tubule. And what that does is that's going to propagate whatever electrical signal is spreading across the sarcolemma. It's going to propagate that electrical signal deep into the muscle cell where it can reach the muscle fibers in there. Okay, parts of a muscle cell continued. The sarcoplasm is going to include myofibrils. There are hundreds to thousands per cell. What these are bundles of myofilaments or contractile proteins enclosed in sarcoplasmic reticulum. And this is what's going to make up most of the cell's volume. Then you have the sarcoplasmic reticulum, which is similar in function to smooth endoplasmic reticulum. It's going to serve as a reservoir for calcium ions, especially the terminal cisternae, okay? It has calcium pumps and voltage-gated calcium channels, the sarcoplasmic reticulum. Okay, so here we have an individual muscle cell. You can see the sarcolemma or the plasma membrane. These holes here are to demonstrate the invagination of the sarcolemma deep into the muscle cell. Which is going to be the T-tubules, okay? Those transverse tubules that dive deep into the muscle cell. These are myofibrils. So if we took a myofibril out and we looked at it, we would see that it's a bundle of myofilaments. So here are the myofilaments right here, the protein molecules themselves. And they're going to make up functional units called sarcomeres. And all of these are going to be packed tightly into this muscle cell. Here you have the sarcoplasmic reticulum that stores calcium, and calcium is going to be really important for muscle contraction. We'll learn about that here in a little bit. There's these terminal cisternae that kind of run down along here. They're slightly larger than the rest of the sarcoplasmic reticulum. They're a good reservoir for calcium, okay? The sarcoplasmic reticulum has pumps that import calcium into the sarcoplasmic reticulum, where calcium is stored, and then release of calcium is going to initiate muscle contraction, okay? So here, showing the terminal cisterna, and you can see it is a storage site for calcium. Okay, so myofibrils, we said, are bundles of myofilaments. So going back, here's the myofilaments, and you bundle them up, you have a myofibril. You put a whole bunch of myofibrils into a muscle cell, and then you put a whole bunch of muscle cells into a fascicle, and then you put a bunch of fascicles together to make up the skeletal muscle, okay? It's a very organized structure, very hierarchical organization. So I want you to be very familiar with it. So the myofibrils are going to contain thick and thin filaments. The thick filaments are made up of about 200 myosin molecules, and myosin itself consists of two strands intertwined with globular heads at the end, okay? Then you have your thin filaments. These are going to be actin, and these look like two intertwined strands of pearls. Actin has troponin and tropomyosin. These are regulatory proteins that are going to regulate muscle contraction, and we'll talk about that shortly. Here you have a myosin molecule showing the intertwining of the two protein filaments, and then the heads here, the protein heads. You put about 200 of those together and you have a thick filament. A thin filament, on the other hand, is just going to be these two strings of actin with tropomyosin and troponin as well. And if you look carefully, there's like these little holes or little pore looking areas on the actin. Those are binding sites for the myosin heads. And normally, tropomyosin covers those binding sites up. So you'll see when we get to muscle contraction, this molecule has to move out of the way before myosin can bind to the actin filament. Okay, so we said the sarcomere is going to be the functional unit of muscle. The sarcomere is an arrangement of thick and thin filaments between a pair of Z discs. Okay, and Z discs are found at the ends of the sarcomere. They're where the actin filaments attach. Here you have the I band. It's light. It contains thin filaments, so only actin in this area. And at the center of the I band is the Z disc where the actins attach. Contrast this with the A band, which is right here. This is going to be darker because it contains both actin and myosin filaments. You have the H zone. This is the central portion of the A band. This is where only thick filaments are present, so there's no thin filament overlap. This area is going to disappear with maximal muscle contraction. So here is the H zone right here. So only myosin in this region. And then down the middle of that's going to be the M line, which is the middle of the H zone, and it's a protein meshwork structure, and it's the attachment site for thick filaments. So basically you have an M line where the thick filaments attach, and you have Z discs where the thin filaments attach. And then these you'll see here shortly, the thick filaments kind of pull the thin filaments inwards during muscle contraction. And here's a better picture of a sarcomere showing the actual structure, like the molecular structure of these proteins. So here's the M line, and this is going to be where these thick filaments attach. You can see the thick filaments overlapping with the thin filaments here, and the thin filaments all attach at the Z disc. The I band is only going to have thin filament. The A band is going to have thin and thick filament, and the H zone is going to have just thick filament. That's kind of in a nutshell what I want you to know about the sarcomere. All right, so here we have an image of a sarcomere. These are going to be your Z discs here. And you can see them highlighted in blue here. The actual myofibril is going this way, lengthwise this way, okay? The sarcomere itself is going to be from Z disc to Z disc. And then you can see the A band. I want you to be really familiar with the A band, which is the dark area, and the I bands, which are the light areas, and what is in each one of those areas, okay? A band is going to be thick and thin filaments. I band is just thin filaments. So muscle fibers are going to have a lot of mitochondria for ATP production. There are myoglobin molecules within the muscle cells that are going to allow for storage of oxygen used for ATP production. So the muscle cells themselves store oxygen to have it ready on hand and ready for when they need to use ATP. They also have glycogen stored, so in their cells for when fuel is needed quickly. And then you've got creatinine phosphate, and what this guy does is it's kind of a holder for phosphate groups, and can quickly give up a phosphate group to help replenish the ATP supply when needed. Okay, now let's talk about innervation of skeletal muscle fibers. So what is a motor unit? A motor unit is going to be a motor neuron and all the muscle fibers it controls. So here is a motor neuron. We have actually two. There's a blue one and a red one. So here is motor neuron 1, the blue one, and you can see it attaches to, or it communicates with these muscle cells in blue. So that's one motor unit. The next motor unit is going to be motor neuron 2, which is the red one, and all of the muscle cells that are supplied by motor neuron 2 are going to be in motor unit number 2. Axons of motor neurons from the spinal cord or the brain innervate numerous muscle fibers. The number of fibers a neuron innervates is going to vary, okay? So small motor units, by definition, have less than 5 muscle fibers that they that they control. This allows for precise control of precise control. Large motor units have thousands of muscle fibers that are being innervated, and this is going to allow for production of a large amount of force, but not very much precise control. Fibers of a motor unit are going to be dispersed throughout the muscle, so it's not just going to be clustered in one compartment like we can see here. These fibers are dispersed throughout. They're not all just clustered right together. The neuromuscular junction. So this is going to be the location where the motor neuron innervates muscle. It has a synaptic knob, which is sort of this swelling here at the end of the axon. It has a synaptic cleft, which is the space between the neuron and the muscle cell. And then it has the motor end plate, which is just a fancy term for this part of the sarcolemma that communicates with the axon. All right, so here we're looking at neuromuscular junction. This is the end of the nerve, or the neuron, excuse me. So this is the synaptic knob. This is the synaptic cleft, that space between the neuron and the muscle cell. And then here's the motor end plate, this whole region right here where all of these acetylcholine receptors are. And this is on the sarcolemma of the muscle cell. So what happens when a nerve impulse comes down the neuron and gets to the end of the neuron, to the synaptic knob, these calcium channels are going to open right here, these voltage-gated calcium channels, and calcium is going to influx. And what happens when calcium influxes, it causes these vesicles of acetylcholine to be released. Now acetylcholine is being dumped into the synaptic cleft, and it's going to bind to these acetylcholine receptors on the sarcolemma at the motor end plate, okay? When acetylcholine binds, that's going to allow for sodium to come into this muscle cell and potassium to leave the muscle cell. And sodium is going to influx really rapidly, whereas potassium is going to leak out much more slowly than that. What happens when sodium influxes is that it's going to change the membrane potential here, and it's going to send an action potential throughout the sarcolemma and down the T tubules into the myofibrils of the muscle cell. And then we will see actual muscle contraction here shortly. Okay, here's the neuromuscular junction at high magnification. You can see the axons here. Here's a skeletal muscle fiber, one muscle cell. And then here is the motor end plate. Okay, so we said the synaptic knob is the expanded tip of the motor neuron axon, and it has the synaptic vesicles that are just sacs filled with the neurotransmitter acetylcholine, okay? When the nerve signal reaches the synaptic knob, calcium channels are going to open up, calcium is going to flow into the neuron, and that's going to be what stimulates the release of acetylcholine. The motor end plate, this is going to be on the skeletal muscle membrane. This is a specialized region of the sarcolemma with numerous folds and acetylcholine receptors, okay? Acetylcholine binds to the receptor, the acetylcholine receptor, and opens sodium and potassium channels, allowing sodium into and potassium out of the muscle fiber, okay? The muscle cell itself. The synaptic cleft is that space between neuron and muscle cell. It's a fluid-filled space that separates the synaptic knob from the motor end plate. And there's an enzyme called acetylcholinesterase that lives there. And that enzyme it breaks down acetylcholine molecules so that you're not constantly having muscle contraction. You need some way to remove that acetylcholine from the acetylcholine receptors, and acetylcholinesterase does that and clears the synaptic cleft of acetylcholine.